Hi, you guys. Excuse me. I want to talk to you a little bit about cell division and cancer. This is the conversation we were going to have before spring break, but I decided to let you go and enjoy your spring break. So I want to do a quick recording of this, and then we're going to start our class on Friday with a little review of this information. So I want to introduce the cell cycle, which is essentially the sequence of events in the life of a cell. So all cells have a life cycle, and the cell cycle kind of defines the sequence of events during the life of the cell. Before we can talk about the cell cycle, though, I need to give a couple terms to us. We're going to use these terms. What we're moving towards in our class at this point is a little bit of understanding around genetics and heredity and all that kind of stuff. So in order to do that, I have to make sure that you understand what DNA is. So I know that we've talked about DNA here and there throughout the course of the semester. But what genomic DNA is all about is essentially, the complete set of instructions in a cell, right? So a cell's complete complement of DNA is called its genome, right? So we can map our genomes now for relatively little bit of money, and that would give us a complete set, right? So we would know completely what kind of genetic information the cells have. In prokaryotic cells, like bacteria, for example, the genome is a single double-stranded DNA molecule that oftentimes forms a loop or a circle. I'll show you a picture in a minute. Sometimes it's also referred to as a plasmid. I believe we used that term previously when we were talking about cells in the beginning of the semester. In eukaryotic cells, however, the genome is basically several double-stranded linear DNA molecules that are bound up and wrapped up around proteins. In the eukaryotic cell, the individual DNA molecules are referred to as chromosomes. So that's kind of what a chromosome looks like down there in the bottom right-hand corner. So again, in these eukaryotic cells, each species of eukaryote has a characteristic number of chromosomes in the nucleus. In humans, human body cells have 46 chromosomes. So when you have two matched sets of chromosomes, so essentially we inherit genetic information from our mothers and from our fathers, and then we get a set of chromosomes, right? When you have two matched sets, so for example, in the human body cell, we have 23 pairs of chromosomes. Each pair of chromosome, if the cell contains pairs of chromosomes, that cell is referred to as a diploid cell. And oftentimes the diploid cell is written as a 2 and the letter N. So 2N is equivalent to diploid cells. If a cell, however, has only a single set of chromosomes, so in the human cell, if you had just 23 rather than the 46, you would be called a haploid cell. And haploid is identified by the lowercase n as in Nancy. So a diploid cell has two matched sets of chromosomes, and a haploid cell has only a single chromosome, or a single set of, or a single chromosome, I guess is what I'm trying to say. So in eukaryotic cells, so down here on this picture down here, this is what a chromosome looks like. And basically, when the cell is not dividing, which we're going to talk about in a minute in terms of the life cycle. So when the cell's in the period of its life where it's not dividing, the DNA is barely visible if you were to look at that cell under the microscope. And that's because the genetic material, the DNA, is uncoiled and it's just sort of scattered about, right? It's loosely scattered about, loosely packed in the cell. When the DNA is starting to get wrapped up around what we call packaging proteins. In this picture, these little purple dots, circles are packaging proteins. They're referred to as histones. Basically what happens is the DNA starts getting wound around these proteins. And that's what chromatin is. Chromatin is just a genetic information that's wrapped around these proteins. And so basically what happens is right before that cell gets to the point of its life where it's going to divide, that chromatin packs up really tightly, and it ultimately will become visible under the microscope. When you can see the chromatin or the individual chromosomes under the microscope, that means that that cell is just about to divide or in the process of dividing. So a couple more terms that we need to know is the term homologous chromosome. A homologous chromosome is simply a matched pair of chromosomes in a diploid organism. So in a diploid organism, like our human body cells, we have 23 pairs of chromosomes, or 46 chromosomes total. So each of those pairs is called a homolog. So the pair is a homologous chromosome. The homologous chromosomes are the same length as their pair. So the two homologs are going to be the same length. They're going to have the same specific nucleotide segments, which are called genes, in the exact same location. So we have genes that code for all of our traits: eye color, blood type, curly hair, straight hair, left hand, right hand, you name it, there's a gene for it. And so homologous chromosomes are chromosomes that are the same length, they're paired, they're the same length, they have the same genes in the same locations on each of the homologs. So for example, in that picture down there at the bottom, we see two chromosomes. Those are homologs. They're the same length, and they have the same genes. They code for the same genes. So even though you see an uppercase and a lowercase, they don't have to be exactly the same, but they're coding for the same gene. Like for example, let's say this is the dominant gene for being right-handed, and this is the recessive gene for being left-handed. So the A would signify a trait, which would be handedness, right or left, and you were going to inherit a gene for being right or left-handed from your mom and right or left-handed from your dad, and those would be found on the homologs, if that makes sense to you. All right, so here's kind of what this would look like. This is if you had the job of lining up the homologs, your job would be to go through, so basically they take out genetic information, they distain it so you can see it, and you line up the pairs, right? They're the same length. In this case, they're the same color, which is nice. They're the same length. They have the same genes for the same traits in the same locations. And you see there's 23 pairs of chromosomes here. This is a human cell. Chromosomes 1 through 22 are referred to as autosomes. They code for all kinds of traits. And then chromosome 23, right here, this is the sex chromosome. So this would be a female. She would have two X chromosomes. If it was a male, there'd be an X and a Y. So those are the sex chromosomes. Chromosome 23 always is the sex chromosome. So 1 through 22 is the autosome, 23 is the sex chromosome. So again, a gene is referred to as the functional unit of the chromosome. So if you look, if you look up here, you're going to see a chromosome, right? And you see that on each of the arms of the chromosome, you have a gene for hair color, for body height, for, in this case, lipoma formation, right? So you're going to inherit two genes for hair color from each of your parents, right? And the gene that you present, the hair color that you see, is referred to as the phenotype. The phenotype is what you see. So in the case of this chromosome and this person, their phenotype is to have brown hair, be tall, and to form multiple lipomas. A lipoma is like a little fatty tumor, right? It's kind of random what genes are found on each of these chromosomes. All right, but a gene is a functional unit of the chromosome, and it's going to determine specific characteristics that are coded for by specific proteins, and those are all found on those chromosomes, right? Traits are just the different forms of the characteristic. Okay, I want you to write down the answer to this on a piece of paper, write the question and the appropriate answer, and you're going to turn this in when you come into class. This is going to give you some participation points. So the first question is, a diploid cell has blank the number of chromosomes as a haploid cell. Does a diploid cell have 1/4, 1/2, twice, or 4 times the number of chromosomes as a haploid cell? So write question number 1, write the question, and write the answer that you think is the correct answer. And I'm going to answer those questions at the end of this video. Okay, second question. I think there's going to be 5 total. An organism's traits are determined by the specific combination of inherited A cells, B genes, C proteins, or D chromatids. An organism's traits are determined by the specific the specific combination of inherited cells, genes, proteins, or chromatids. All right, so let's quickly look at the cell cycle. The cell cycle, again, like I said previously, is just a series of events that essentially involves the cell's growth period, its life, and then finally its division period if it's a dividing cell. If it is a dividing cell, we're going to start with one cell, and it's the cell cycle. Once the cell divides, it's going to produce 2 new daughter cells. And the important thing to remember about this is in this example of this type of cell division, the daughter cells are genetically identical to the parent cell. So you start with one cell and you essentially replicate it. You make an exact copy. The cell cycle is timed and it's very carefully regulated. This is what the cell cycle looks like. Right? This is a picture out of the Concepts of Biology book that you have free online. So you see the cell, so this is the life of the cell. So this is the growth period of the cell, the part where the DNA is made and copied, and then the finishing portion of cell growth. So if you look here, we have in gray, the first part of the cell cycle is called the G1 phase. The second part is called the S as in Sam phase. The third part of the cell cycle is called the G2 phase. But the overarching name for all 3 of these phases, G1, S, and G2, is interphase. So interphase is not part of cell division. It's the part of the life cycle that the cell does whatever it needs to do, whatever its job is. It grows, it develops. The DNA ultimately is going to get synthesized and copied, and then it's going to wrap up the cell division, the cell growth phase at the end here. And then if it's a dividing cell, it's going to go through this portion of this cell cycle, which is called cell division. In this type of cell division is referred to as mitosis. This is where we're going to form 2 daughter cells. All right, so the cell cycle is very timed and it's carefully regulated. So essentially there's basically 2 major phases of the cell cycle. One is interphase. So all of this purple, this blue, and this kind of grayish color, all of this is interphase. And then this little piece of the pie here, kind of in that coral color, is mitosis. So again, interphase. During G1, the cell's active at its biochemical level. It's doing whatever it does biochemically. Some cells make proteins, some cells make enzymes, some cells, do all kinds of stuff. It's doing it during G1. Then, the important thing to remember at the S is in Sam, in the S phase, this is where DNA replication occurs. Remember, what we're talking about here is we're starting with one cell and we're ending with 2 cells if it's a dividing cell. If you start with one cell and you end with 2 cells and those cells are exactly the same, then before you can start with one cell and divide it up, you've got to double everything in there. So essentially all the genetic information gets copied and then it gets divided, and then we have 2 cells that are exactly the same. So when the DNA replication occurs, basically what that's going to result in is 2 identical copies of each chromosome. And when you have 2 identical copies of a chromosome, we call those copies sister chromatids. And they're joined together. So they're joined together by a little kind of like a button, if you will, in the middle, and that thing is called a centromere. I'll show you a picture in a minute. All right, so G2 is the last part of interphase, and that's where the cell replenishes its energy stores, and it makes the final preparations for cell division. So whatever has to be wrapped up in order for that cell to actually divide, all of that gets wrapped up in the G2 phase, which is still a part of interphase. And then we can go into mitosis. So that's what's going to happen next. We know that there's all kinds of signals that are going to tell the cell when to divide, when to stop and repair if something got screwed up in the DNA copying. Maybe it got irreparably damaged and we need that cell to die. Or sometimes the cell goes into what we call a dormant phase, which is a G0 phase. That's a cell that's not going to divide. It would go into G0, right? It would skip mitosis and just kind of get banked, right? Kind of just get held up. All right, so here are the phases of mitosis. I'm going to kind of go through this quickly. We're going to do this in the lab this week, and you're going to get some practice with kind of putting this in order for yourself. So the phases of mitosis, this is cell division, gets divided up into basically, I kind of want to think of it as 4 phases. I'm not going to focus on this pro-metaphase portion here. So during the first part of cell division, the first phase is called prophase. The important things that happen during prophase are all written down here. So what's going to happen during prophase? So the chromosomes are going to start to, in the nucleus, are going to start to condense if they haven't already done so, and they're going to become visible. Then what's going to happen are these little things here that are kind of orange. These things are called centrioles. They're part of the cell, the cytoskeleton of the cell. The centrioles are going to split, and they're going to migrate away from one another, and they're going to go to opposite ends of the cell, like you can see happening here towards the end of this phase. And then what's going to happen is they're going to spit out these little fibers, which are called spindle fibers. And those spindle fibers are ultimately going to go and attach to each one of these sister chromatids that are the duplicated chromosome, that happened during the S phase. The other thing that's going to happen is the nuclear envelope, or the nuclear membrane, is going to dissolve all the way by the end of prophase. And the spindle is ultimately going to go and attach to those centromeres. Okay, then we go to the next phase, which, again, this is kind of a continuous thing here. So let's talk about our second phase that we are responsible for knowing, which is metaphase. The important thing about metaphase is, okay, the nuclear envelope is gone. The thing that happens during metaphase that makes it very easy to identify is those sister chromatids that are joined together at the centromere. They line up right down the middle of the cell. They call that the metaphase plate, but it's just basically the middle of the cell. Each of those sister chromatids are attached to the spindle, right? So one's attached here, the other one's attached here. And that's important because when we go into the next phase, which is anaphase, those sister chromatids are going to get pulled apart, right? They're just copied. So half of them are going to go this way, half of them are going to go that way. That's happening in anaphase. The cell starts to elongate during anaphase. And then we go through our fourth phase, which is telophase. The nuclear envelope starts to reform around the genetic information on these two kind of clusters of genetic information. The spindle breaks down. The chromosomes are kind of at the poles. And then what's going to happen is this whole thing is going to elongate, and it's ultimately going to start to pinch off. When the nuclear envelopes are totally reformed, everything left that's in the cell, the cytoplasm and whatever else that's in the cell, is going to get divided and separated, and we're going to end up with 2 new cells. So telophase and cytokinesis are kind of a continuum. Cytokinesis ultimately, and then an animal phase, they call this a cleavage furrow, where it sort of gets squished in the middle, and then ultimately they pull apart. And those 2 cells, those 2 new cells, are exactly the same as the parent cell that we started with. So again, that process of pulling those cells apart, animal cells do it a little bit different than plant cells. Plant cells make a cell wall, right? Plant cells have both a cell membrane and a cell wall. So they put this plate down in the middle, and then you kind of pinch off the 2 cells like this. The animal cell creates, what we call that cleavage furrow, and then they pinch off. But in both cases, the cytokinesis signifies the completion and the physical separation of all the components of the cell that are in the cytoplasm, resulting in 2 new cells. So I want to show us talk a little bit about G0. So not all cells divide. For example, cardiac muscle cells, the cells that make up the walls of your heart, do not divide. Nerve cells do not seem to divide. And so instead of going into mitosis, they go from G1 into what we call a G0 stage. They're not preparing for cell division, and they're going to stay in G0 permanently for the entire life of that cell. Prokaryotes do this differently. They do it faster. Remember, the DNA situation in prokaryotes are different. That's kind of the single loop of genetic information. Prokaryotes divide by a process, called binary fission, where essentially they just replicate like crazy and just divide and divide and divide and divide. It's a lot faster. They don't have to go through all those processes, right? No mitosis in prokaryotes, just binary fission. We'll talk more about binary fission when we talk about bacteria later on in the semester. Okay, so I mentioned there's some regulation of this cell cycle, and it makes sense because we don't want mistakes, right? So when we have mistakes in either the duplication or the division of the chromosomes, that leads to things that we call mutations. And once the genetic information has been mutated, and that cell, if it continues on the process of division, then that mutation is going to be passed on to every new cell, right? So basically we're going to produce abnormal cells from what would have been a normal cell. And so we've got these checkpoints put in place to kind of make sure that this all goes correctly. The first one is the G1 checkpoint, and that just checks in to make sure that the G1 stage went okay and everything is okay. The DNA is okay. It's not damaged, because remember what happens here in the S phase is the DNA gets replicated. So if it's damaged, we don't want to copy it. Then at the end of the S phase, we've got another checkpoint. That's the S phase checkpoint, again, to check in on the DNA, making sure it's replicating properly. Then we've got our third checkpoint, which is the G2 checkpoint at the end of the G2 phase, which is that secondary growth phase. What happens with G2 is we check in to make sure that all the DNA got replicated, that if there was damage to the DNA, it's reparable, right? It can be repaired, and that everything is in place that we need to be in place in order for division to occur, specifically that spindle-making apparatus, right, from the centrioles with those long fibrous kind of cords. And then if the cell does start to divide at the termination of metaphase, there's another checkpoint, and that's called the metaphase checkpoint. It's going to check in to make sure the spindle is built correctly, that all the chromosomes have spindle attached to them so they can be pulled apart, and that the chromosomes are lying down right on that metaphase plate or the equator of the cell. So if there's anything that doesn't check out here, sometimes that will signal the cell to actually die, and that process is called apoptosis, which I'll show you on a slide in a minute. So again, mistakes in the duplication or division process leads to things that we call mutations. We're going to talk some more about that when we talk about genetics. And when there is a mutation, that mutation, if it doesn't cause the cell to die, that mutation then would pass on. And now that we are able to sequence the genome for so many people, and any of us could do it, we can find mutations in our genome pretty easily. And not every mutation is incompatible with life. Most aren't. Not all of them even present in population, but the presence of them tells us that there was something going on with the DNA copying, most likely, or something in the cell cycle, one of those checkpoints. Where we would have stopped, we kept on going. And I should say that evolution is essentially mutations in action. So not every mutation is incompatible with life. Most of them aren't, right? A lot of them aren't a big deal, but the ones that are, we hope to catch up to. All right, so again, the term for programmed cell death is called apoptosis, and the reason for this is to weed out cells that should say harm, weed out cells that might harm an organism, or eliminate excess cells, and in order to carve out functional structures. So let me show you an example of that. So here we have this cute little chick, right, and the development of the little feet of this cute little chick. So in utero, right, the little feet are webbed, right, and so essentially what happens is, and this happens in humans too in utero, our little hands and feet are webbed, and so what basically happens is apoptosis is stimulated in the cells in between the individual little talons, I guess we'll call them. And what that would ultimately do is it would cause these cells to die, and then it would just cause the individual little chick fingers to be there, right? So that's perfectly normal, right? In the process of fetal development, there's a lot of programmed cell death. All right, so it's not always a bad thing. It sometimes is a very good thing, but it's also very beneficial if we do have some kind of mutation that would definitely harm the organism, that would get caught, and that cell would not proceed to divide. If we lose control of the cell cycle, that's essentially what cancer is all about, right? So we know that we have these go signals and stop signals all the time, kind of trying to keep things balanced, trying to keep cells under control. One thing we know about tissues is there's groups of cells that are all working together, same kind of cells working together to perform a function, right? And so they all kind of respect the boundaries, right? If the stop and go signals are in balance, then the tissue is not going to divide too much. If the go signals go crazy and the stop signals are less intense, then what will happen is this tissue will divide and divide and divide rapidly, and you'll end up with a tumor. That's what a tumor is, essentially. It's an overgrowth of cells. And so that's what cancer essentially is in a very oversimplified way. It's an abnormal mass of tissue. And that essentially is the result of the loss of control between cell division and cell death. So in this terminology with tumor, there's two different ways to think about tumors. One is a benign tumor, which traditionally is slow growing and usually relatively harmless. Not always, but usually relatively harmless. So it's this mass of abnormally dividing cells that's not dividing super, super fast. And the other thing about benign tumors is the cells tend to stay together. They tend not to like get thrown off and go to someplace else and start dividing like crazy right there. A malignant tumor, however, is a tumor that grows very quickly in most cases, and it doesn't stay together, so it starts to invade surrounding tissues. The term for the invasion of surrounding tissues by an abnormally dividing cell is called metastasis. So when a tumor metastasizes, what it does is it sends that abnormal cell to a new place, and it starts dividing like crazy there. So it's a little bit tricky with cancer because cancer cells are not normal cells, but they don't look necessarily abnormal to our immune system, right? Like if we were to get a bacterial cell in our body, those cells look very different. They're different types of cells, so our immune system recognizes it. Cancer cells, in terms of the surface of the cells, they have the same kind of surface markings. So they might look different, but your immune system doesn't always catch it. So some kind of characteristics of cancer cells is they look a little bit different. They essentially are immortal, and that's because they don't respond to signals to die. They produce their own signals, which makes them divide faster and faster and faster. They lack what we call contact inhibition, which means normally when a cell butts up against another cell, that inhibits its growth. They lack that. They're like the bad neighbor. They just keep putting more garbage on the lawn, if you will. I mentioned already that they don't undergo apoptosis when they're damaged. And the other thing that's really interesting about them is they send signals to stimulate the growth of new blood vessels. And the reason why they do that is because they are dividing so quickly. They're very metabolically active and they need a lot of resources. They need a lot of nutrients. And the way we get all of our cells good nutrients, as we know, is from our blood. So basically what they do is they send out these signals to grow their own vascular supply. And the fancy word for that is called angiogenesis. So they're not normal cells, right? We have this imbalance between our go signals and our stop signals. We all have this going on all of the time. We have these genes. The go signals are produced by these genes called oncogenes. The stop signals are produced by these genes called tumor suppressor genes. We have them, and most of the time they're in balance. So we don't have these tumors growing all the time. But if anything happens that kind of messes up those signals, then a tumor or a cancer can result. Some of the things that we know that can alter DNA and cause mutations that could ultimately lead to cancer formation are harmful chemicals in our environment, toxins, environmental toxins. Radiation is a known carcinogen. Sometimes viruses are very tricky, and we'll talk more about them, but they basically hijack our DNA, and they could make they basically cause the cells to start producing viral DNA, which is kind of messed up. Lifestyle factors can attribute to this: poor diet, lack of exercise, sun exposure, especially UV radiation, and smoking will increase the risk. We'll talk some more about all of that when we talk about genetic mutations and epigenetics. The treatments for cancer traditionally are surgery. some combination of chemotherapy, which is drug therapy to be targeted at rapidly dividing cells, of which cancer cells are. Radiation will kill rapidly dividing cells. Drugs that inhibit this formation of these new blood vessels are kind of newer on the scene, and they're called angiogenesis inhibitors, and also drugs for the immune system. So in a nutshell, that's as simplified as we can make it, but that's kind of what goes on with cancer. All right, so finish this up. Again, you're going to bring this into class with you. Chromosomes are duplicated during what portion of the cell cycle? When are the chromosomes duplicated? Is it during the G1 phase, the S as in Sam phase, prophase, or prometaphase? And separation of the sister chromatids is a characteristic of which stage of mitosis? So just write the even if you don't know, write this question down and go back and think about it with your notes. Is it prometaphase, metaphase, anaphase, or telophase? I think there's one more. What is necessary for a cell to pass the G2 checkpoint? The G2 checkpoint, right? Has the cell reached a sufficient size? Does it have an adequate stockpile of nucleotides? Has it accurately and completely replicated the DNA? Or has proper attachment of mitotic spindle fibers, I think that should say to the sister chromatids. I've stopped typing there. Okay, the G2 checkpoint. Proper attachment and formation of the spindle. Let's just say that. That will work. Okay, so that concludes this. Make sure that you watch it, review it, bring your notes, because we are going to start with a mitosis exercise on Friday. See you then.